the branch of genetics which deals with the study of chromosome and its role in transmission of trait from one generation to the next generation.
Correct. You are right. You have correctly defined the term cytogenetics. And in cytogenetics, we have to study the Mendelian principle of inheritance, the linkage, and post-Mendelian development. Post-Mendelian development, in that section, you have to study the interaction of gene and various and interactions of genes, and various types of interaction of genes will be dealt with. So in Mendelian inheritance, you will study linkage, you will study. You will also study the interactions of genes. You have to study structural and numerical changes in chromosome. That is the numerical changes. Numerical changes in chromosome is known as the genomatic mutation. Similarly, the structural changes in chromosomes changes in chromosome constitute the chromosomal aberrations. And beside this, you have this genomatic mutation is of two types: the aneuploidy and euploidy. And these are very important topic also from the examination point of view. So this cytogenetics is a very broad area included in your syllabus. So in the just because of we have to study the Mendelism, the principle of inheritance, and I have already told you that to for a better understanding of the Mendelian principle of inheritance, you must have a very sound knowledge of the chromosome and meiosis, because if your concept is not clear with respect to the chromosome and the meiosis, it will become somewhat difficult to understand the Mendelian principle of inheritance in a better way. So I have also advised, I had already advised you to go through the books, which even if you do not have the books of the PK Gupta, you may go through your NCRT book for a quick look, and and you can revise whatever have you studied about the chromosome in your NCRT books and meiosis, so that you can easily understand the principle of inheritance. I know that you might have studied the Mendelian principle in your previous class, like in 12. But in BS at this level, at BSc Biotic Part 1 level, you have to understand in a very broad sense what actually Mendel was interested to convey out of his hybridization work done. And you all know that the Mendel's law of independent assortment, and Mendel's law of dominance, and law of segregation. And but you know that law of segregation, which is also known as the law of purity of gamete, is universal. And law of dominance and law of interaction and law of independent assortments are not applicable in all the cases. There are so many exceptions to these laws. So just as per my suggestion, I think that you are quite aware of the significance of your better knowledge of the chromosome and the meiosis. So in the just prior to starting the Mendelian principle of inheritance, I have started to deliver lecture. I have started the topic chromosome, and in the last two lecture, I have discussed about the chromosome, its structural organization, and various parts of the chromosome, like the telomeres, centromeres, and chromatids, chromonema. And these points have also been dealt with while I was teaching the differences between the prokaryotic and eukaryotic cell, because you know that eukaryotic organisms have the chromosome, and in case of prokaryotic organisms, the chromosome like structure is not found. Rather, the bacterial genome is present, consisting of the DNA in case of bacteria. So the chromosomes is a very important topic you have studied. So this chromosome, in the last lecture, I have described the heterochromatin and euchromatin. This heterochromatin, I think that you have already studied the differences between the heterochromatin and euchromatin. Can any one of you tell me some of the points which I have taught you? Anyone? 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 Just I want to recall, I want to see whether all the points are in your memory or not.
Yes, sir.
Point out.
सर यूक्रोमेटिन में लाइटली स्टेन्ड पार्ट ऑफ क्रोमोसोम यूक्रोमेटिन लाइटली स्टेन्ड पार्ट ऑफ क्रोमोसोम होता है, बट हेट्रोक्रोमेटिन डीपली स्टेन्ड पार्ट ऑफ क्रोमोसोम होता है। यूक्रोमेटिन में एक्टिव जीन्स प्रेजेंट होते हैं और हेट्रोक्रोमेटिन में इनएक्टिव जीन्स प्रेजेंट होते हैं, वो जेनेटिकली इनएक्टिव होते हैं।
Yes. What are the where? Let me know something about their location. Where the euchromatin, where the heterochromatin is located along the length of chromosome. Which part of
सर
the chromosome is heterochromatin? Heterochromatin.
हेट्रोक्रोमेटिन सर टेलोमेरिक रीजन में होता है
Yes.
और यूक्रोमेटिन पूरे क्रोमोसोमल लेंथ में होता है।
ठीक है. The very important, you are missing something, missing some points. I had mentioned you that the heterochromatic part is mostly the telomeric part is made up of the heterochromatin. So the heterochromatin is located in at the telomeric, and the very important point where the telomere is located is the around adjacent to the centromere. Centromere in near centromeric just near centromere. Centromeres center just around the centromere, the heterochromatic zones are present, and you have correctly you have pointed out all the very important feature of the heterochromatin. And it is, you know, that I have also taught you about the types of the heterochromatin, which include the constitutive
Yes.
heterochromatin and facultative heterochromatin. What are the differences? What is the main difference between the constitutive and facultative heterochromatin? Let me know.
Yes, sir.
Sir, constitutive heterochromatin is that type of heterochromatin which contains highly repetitive part of DNA.
Yes. And what is facultative heterochromatin?
It does
Facultative
not
is
remain heterochromatic. But yes,
Facultative heterochromatin?
facultative is
hetero-
actually euchromatin, but sometimes it behaves as heterochromatin.
Sometime you are correct. You should say like this, that at sometimes what happens, the euchromatic part of the chromosome undergoes heterochromatinization by undergoing condensation, coiling and recoiling to become very condensed part.
Yes, sir.
And so such facultative heterochromatin is actually the euchromatin which has undergone condensation and coiling and recoiling to form the condensed part. And this process is known as the heterochromatinization. And I had also cited one of the very important examples of the heterochromat, such a structure which has been formed by the heterochromatinization. And I think that you might be knowing that. I think that it might be in your memory. The
Yes,
best example, what is the example?
sir. Bar body. It's bar body.
Yes, and this bar body is the product of the heterochromatinization of one of the two X chromosome found in the somatic cells of the human female. Because in male there are because in female there are two X chromosomes, and in male there is only one X chromosome. So one of the two X chromosome found in the human female cells undergo inactivation, and this inactivation, how does this inactivation takes place? This inactivation takes place by the coiling and recoiling and condensation of the uh, X chromosome, and it becomes a dot, and it appears a dot like a structure. And this phenomenon and this process of the inactivation of one of the two X chromosome is known as, what is the term used for that? Can you tell?
Lionization.
Yes, lionization. And the
their
very
doses
important,
compensate. Their
yes,
doses
this is very important
compensation.
point. Dosage compensation. Dosage compensation, because the dosage, or because the dose of the gene present on the X chromosome, present on the one chromosome of the male, okay, but in case of there are two X chromosomes, so the dose of the genes present on the X chromosome should be adjusted. If the one of the two X chromosome does not undergo heterochromatinization, what will happen? The female cell will be having just the double number of genes in comparison to the male partner. So this process, therefore, this is also known as the dosage compensation. So this dosage compensation, this process, this phenomenon was first reported by the lion, and therefore this process is also named after the discoverer of this process and lionization. So this is one of the, this bar body is one of the best example of the facultative heterochromatin. So you know that these heterochromatin and euchromatins are very important topic as far with examination, as far your examination is concerned, from the examination point of view, because and frequently you are asked to write short notes on the heterochromatin, and you may be asked to distinguish between the heterochromatin and euchromatin. So today I thought, yesterday I thought uh, that I must have an interactive session with the student, because you know that online teaching includes the delivery the delivery of lecture by the teachers on the topic of on the topic, interaction with the students, testing of the students, and evaluation of the students. So in this lecture I am just interacting with you, and I decided to interact with you what on the topic whatever I have taught early in the previous lectures. And I think that some of you are very are taking interest in interacting with me, and some of them are not. Why some of you are not taking active part in this interaction, teacher student interaction? Why? What is only one student is responding to me by answering the question which I ask, and another are only listening, other are only acting as the listener only. This is not a good sign. All of you must interact, and if you have any problem in understanding the topic, you may ask me. Okay, but I expect from you that you must interact with me on the topic. This interactive session makes the lecture very, makes the online teaching very fruitful. What is kinetochore? Anyone? What is kinetochore? I have already taught you the chromosome. What is kinetochore?
Yes.
What is
Yes, sir.
kinetochore?
The division of nucleus.
No, kinetochore. What is? Let me know that there is a structure uh, as present on the chromosome, and known as kinetochore. So what is, where is kinetochore located, and what is kinetochore?
As page.
ये क्रोमोसोम के मिडिल पार्ट में होता है, वो दोनों क्रोमेटिड्स को अटैच करके रखता है डिस्क लाइक.
You are making you are trying to answer, or but I think that there should it requires some rectification. I want to rectify you that this kinetochore is a 3-layered protein disk. This kinetochore is a 3-layered protein disk associated with the primary constriction centromere. You will have, this is the correct answer. The kinetochore is a 3-layered protein disc associated with the primary constriction, and you know the difference between the